Welcome back, friends. This is part 3 of our TCBS Agar Video Series for shrimp aquaculture. In part 1, we learned the importance and preparation of TCBS Agar. In part 2, we learned how to plate water and post-larvae samples. In this video, we will understand the principle of TCBS Agar, different colony colors, colony morphology, and how these observations help us judge pathogenic and non-pathogenic Vibrio load in aquaculture systems. First, the principle of TCBS Agar. TCBS stands for thiosulfate, citrate, bile salts, and sucrose agar. It is a selective and differential medium mainly for Vibrio species. High alkaline pH, bile salts, and citrate suppress gram-positive bacteria. Sucrose with a pH indicator bromothymol blue differentiates bacteria based on acid production. Always remember, do not interpret TCBS plates based on color alone. Colony morphology, size, shape, margin, surface, and density is equally important. Now let us start with yellow color colonies. Yellow colonies are produced by sucrose fermenting Vibrio, such as Vibrio alginolyticus, Vibrio fluvilis, Vibrio fornesi, and some non-O1 Vibrio cholerae. Typically, less pathogenic yellow Vibrio form medium to large, smooth, round colonies with uniform yellow color. When yellow colonies are dominant, it usually indicates high organic load, excess feed, or plankton crash. These act as an early warning, not always a disease condition. Next, green or blue-green colonies. These are produced by non-sucrose fermenting Vibrio, such as Vibrio parahaemolyticus, Vibrio harvey, Vibrio vulnificus, and Vibrio campbelli. Pathogenic Vibrio usually form small to medium, dense, dark green colonies with well-defined edges. High numbers indicate serious disease risk. Based on colony morphology, we can get a presumptive idea of pathogenic dominance. Important caution: not all blue-green colonies are Vibrio. Bacteria like Pseudomonas and Aeromonas can also appear blue-green on TCBS and need further confirmation. Now about black color colonies. Black colonies are usually produced by hydrogen sulfide-producing bacteria, mainly Shea-vanilla. They show black or dark centers and are common in sediment and gut samples. They indicate anaerobic conditions and organic accumulation, not primary Vibrio infection. Now pinpoint colonies. These tiny colonies are often produced by Proteus, Enterococcus, Aeromonas, or stressed bacteria. Pinpoint colonies should not be counted as Vibrio and usually indicate background contamination or stress. Now let us look at common Vibrio ranges in aquaculture samples. In hatchery or farm water, ideal Vibrio count is below 100 CFU per milliliter. Counts between 100 and 1000 require close monitoring. In culture pond water, Vibrio counts below 1000 CFU per milliliter are acceptable. In pond sediment, Vibrio counts are naturally higher, usually between 1000 and 10,000 CFU per gram. In shrimp gut samples, normal Vibrio counts range from 1000 to 10,000 CFU per gram. Higher counts with pathogenic morphology indicate gut infection risk. In shrimp haemolymph, Vibrio should not be present. Even a single colony indicates septicemia and high mortality risk. Always remember, TCBS Agar gives presumptive identification only. Final confirmation requires biochemical or molecular tests. But for routine hatchery and farm monitoring, color plus colony morphology plus CFU provides a very strong practical diagnostic tool. This completes part 3 and the final part of our TCBS Agar video series. If this video was useful, please like, share with farmers and hatchery technicians, and subscribe to the channel for more practical shrimp aquaculture microbiology content.